Good afternoon to one and all. Welcome to the sixth webinar in the webinar series Emerging and Re-emerging Viruses. We started this webinar in May 28th on every Thursday. We had a speaker in the area of emerging and re-emerging viruses who used to give a good talk on the relevant topic in the week of COVID-19 epidemic. Today we have Dr. Rajendra Pilankatta. He's giving us a talk on the topic relevance of immune enhancement during the emergence of COVID-19 pandemic. Introducing the speaker, he's an associate professor in the Department of Biochemistry and Molecular Biology, Central University of Kerala, Kasaragod. After doing his PhD from Osmania University, Hyderabad, he worked as research associate at International Center for Genetic Engineering and Biotechnology in New Delhi, and worked as postdoctoral researcher at Harvard Medical School, Boston, US. As a C.V. Raman Fellow, Dr. Rajendra also worked in the Department of Microbiology and Immunology at Georgetown University, USA. Currently, he is an expert member at the Institute of Advanced Virology, Trivandrum. His work is mainly on viruses and immunology-related area. He has over 33 publications in peer-reviewed journals to his credit. On behalf of KCST and all participants joined us for the webinar, I welcome you, sir, for the presentation. Over to you, sir. Thank you.
Thank you, sir. Thank you for the nice introduction. Good afternoon to all. First of all, I would like to extend my thanks to KSCST as well as Institute of Advanced Virology members for giving me this opportunity to present a topic in the emerging and re-emerging viruses. Actually, hope some of you will be knowing, since the last 3.5 months, we are working for the diagnosis of COVID-19 in our laboratory. In fact, our research lab, Department of Biochemistry and Molecular Biology, the entire team members of the department actually actively involved in the testing of COVID-19. So currently, entire district sample we are testing here. So, in fact, I am not an expert in the COVID-19 and SARS viruses, but my major area of work is in dengue viruses. And of course, both have a common thing, that is a positive strand RNA viruses, these are. And in fact, in the immunopathogenesis also, there are some certain kind of overlap also. So I thought of sharing some of my thoughts in this event regarding COVID-19, emergence of COVID-19 pandemic, with respect to some of my area of interest of research, what we are doing currently in our lab here. So before going to detail of immune enhancement, let us have a brief background. We know that human population have experienced major epidemics. Historically, it has actually changed the course of human history itself. If you really go through the history, we can see that the smallpox in 1500s actually caused actually resulted in the ending of Aztec civilization, even Pacific civilization. Entire one particular civilization itself, it actually doddered away. Completely abolished. That's what history tells us. So the pandemic infections, actually communicable diseases, infectious diseases, mainly viruses, and bacteria, they can do unimaginable damage to the community and for the entire human race itself. And if you go back again in the 14th century, if you see plague, everybody knows that. Actually, European population, a third of the European population over 5-year period, completely eliminated. So we have to see these things are happening before globalization itself. Now we are in an era of globalization. the travel within a second, within hours, we are reaching any corner of the country. So there is a lot of things we actually connect in this regard. Now, when we come back to the middle of the last century, 20th century, we actually reached a kind of comfortable zone. I mean, where we thought we can control the many infectious diseases, and in fact, in the developed countries, the community in the developed countries, they felt like, I mean, we can actually completely control the mortality, but this perception actually is not, I mean, it's not a sustained
Hello.
perception what we are having. That's what I told. Can we eliminate viral infectious disease completely? I can say it is not possible completely elimination. If one is going, another is coming. That's what history is telling. And curbing of smallpox really strengthened the perception that infectious disease could be eliminated. The reason was for that actually the vaccine. We know that the actual name comes from the vecca in the based on the vaccine. Vecca means cow. You know that story how vaccine is developed. And also a lot of especially in the second half of the last century, a lot of development has happened in the basic life science and physics and chemistry and drug development, drug screening, genome sequencing. A lot of milestone events happened. I mean, so we can see that the contribution of all these development really helped in the discovery of many antimicrobial agents, and also vaccine. Also, people are they started to use the more advanced method than the traditional method. So that was one confidence what we got in, we can actually control this kind of infection. And again, another thing is by that time, especially in the developed countries, better sanitation, clean water, better living conditions has evolved. The thing is, but when we were continuing in that quasi kind of zone, I mean, we can feel that old world communicable diseases are slowing down, but the new things are coming up. New things are continuously posing challenges against the human race itself. Now we know that a new unknown set of viral infectious disease actually started to emerge with the newer name, and sometimes it comes in the name of the place and many other things. So we know that we are hearing avian influenza and swine flu, and finally the SARS coronavirus and MERS virus and SARS-CoV-2 and also Ebola, Nipah, dengue virus, chikungunya. I mean, so on. A big list is in front of us. It is emerging. The thing is now whole world is in the pandemic grip of SARS-CoV-2. So then what is actually by definition emerging and re-emerging viruses? The emerging viral infections actually, the viral infections whose incidence has increased within the recent past actually. I mean, we can, by the situation, by the context actually, we can even think that it will increase in the near future. That kind of infection we are actually putting under the classical definition. It's emerging viral infection. But re-emerging viral infection is actually reappearance of a viral infection, which was once endemic and we controlled, and again re-emerging, probably because of the finding of a newer host adaptation, genetic variation, so many factors, due to so many factors, so it can re-emerge. So the why this re-emergence and emergence is happening, and we can see there are as I told, in the last century, in the second half of the last century, actually, we can see the thing is in the especially developing countries, and the population explosion and poverty, malnutrition, and increased domestic and global connectivity. These are all became the helped actually supporting factor for the emergence of newer and newer viral infectious disease. Economic factors also leading population migration. So we know that the globalization is again one of the main thing, and we know actually after the Second World War, there were a lot of cargo transportation, and similarly the microbes also got translocated to different areas of the globe. infection. So the social practices, prevalence of immunosuppressive diseases, and another important thing, very, very important thing is the developing countries, it's a process in development. The unplanned urbanization was one of the really major cause for the, especially certain kind of, arboviruses and many other viruses. We will talk about that very soon. I mean, unplanned urbanization is a big cause, and deforestation. Deforestation, actually, the deforestation, when we do, actually we are affecting the habitat of many small animals to larger animals. So many of the viruses find their reservoir in the animal kingdom. So the moment we encroach their habitat, what happens is it retaliates in very bad shape sometimes. I mean, we will see emergence of certain newer infections. So, of course, the genetic alteration is of the pathogen is also very, very important. So, it's a very complex phenomenon why viruses are emerging and re-emerging. All these factors are contributing for this. And the thing is, when you're coming back to the, I mean, again, the beginning of this 21st century, actually, this century itself, within 2 decades, we are now seeing 3 important epidemic, and also one. Now we are in the pandemic situation. The first one is it started actually in the 2002. That is SARS coronavirus. SARS coronavirus is actually, again, the origin is from China, and it spread within few months. It spread many places, many countries, and around 8,098 individuals became sick, and 774 deaths happened. And if you compare, the mortality rate is relatively higher when you compare to the SARS-CoV-2. In the case of SARS-CoV, it happened in 2002. And similarly, again, MERS-CoV, Middle Eastern Respiratory Syndrome viruses, that is in 2012. There you can see around 858 people died, individuals died, and their actually mortality rate is high. Case fatality rate, what we are calling number of deaths, actually is more in the case of MERS-CoV. I mean, but now our situation, we are whole human race in the pandemic grip of SARS-CoV-2. So in the slide, you can see 5,07,000 is as on June 30, but today it is 5,16,000 across the world. So then what is this coronavirus actually do, and how, from where it comes, genetically what it is? Actually, as I told in the beginning, it is a positive strand RNA virus. Actually, coronaviruses are positive strand RNA viruses. It's a quite bigger when you compare to dengue and all. What we are 11 kb size, and this is actually around nearly 30 kb size. The important thing is clinical symptoms of human coronavirus infection ranges from simple common cold to severe pneumonia and bronchiolitis. And common cold, we were actually the coronavirus infection. We know about coronavirus since 1960 actually. 1960 onwards, we are living with the coronavirus. I mean, earlier also we would have been there, but in the literature, what we are finding is from 1960. These are all called, we were thinking it is a nuisance virus, and where it's just causing a simple cold. So we may ignore that. I mean, simple cold and it recovers. But the respiratory infection it causes a lot of damage in the global scale every year. I mean, in the flu viruses. So in this, if you see, coronavirus, it belongs to family Coronaviridae, and as I told, it's a positive single-strand RNA virus. And the subfamily, in the subfamily Orthocoronaviridae, we can find that there is a beta coronaviruses and alpha coronaviruses. In that, what we are seeing is in the beta coronaviruses, many of them already known to infect humans, and that is the first one, HCOV, OC3, and HKU1, and finally, these are now familiar names. I mean, SARS-CoV, that is happened in 2002, and MERS-CoV, that's happened in 2012, and then SARS-CoV-2 now in 2019-20. And this is, in this group, actually, it is there in the actually origin is from bat, actually. So similarly, we can't forget one in 2008, there was one major epidemic in the livestock, that is actually again in China. that is severe acute diarrhea syndrome viruses. And so you can see that gamma coronaviruses are there again in the other side, and delta coronaviruses also are there. So what we are actually in the pandemic grip of actually beta coronavirus, that is SARS-CoV-2. So let us see how this actually transmitted between the people. I mean, we know that it is a communicable disease by contact, by aerosols. So you can see in this figure, the indirect or direct contact can cause the infection. The primary site of infection is the respiratory tract, and so it is you can see that droplets and also coughs, sneezes, aerosols, saliva. All these secretions can transmit the viruses between the people. And this is what actually we are calling the breaking the chain. The chain is should be broken here, where it should not transmit between the people. And possible routes of transmission, whole blood serum also. I mean, and you say what I'm telling is a COVID-2 transmission. What we are telling based on our knowledge on SARS-CoV, which happened in 2002. SARS-CoV areas are there still. We have to do a lot of research to understand that. And also urine and breastfeeding also, it is actually reported contact with the contaminated surfaces. That is the one thing where breaking the chain is very important. And now we are coming to these three SARS viruses, where it was and how come it is now it is coming to human being. I mean, earlier where it was. When you see this, you can see the intermediate host is there in between. Origin is virus. Origin of the coronavirus, SARS-CoV-2 virus is actually having a similarity with the SARS-CoV more, and more than this, it is very close to bat coronaviruses. So the bat coronavirus is actually closely related to the origin. We know that it is from bat, and directly from bat to human being, it is not spreading. So there are some intermediate hosts. How it is coming intermediate host in between, that's what I was telling. So how we are interacting, how we are worshipping our nature. In other words, I mean, the way our habit, all this matters. So intermediate host for the SARS coronavirus 2003 is civet cat, and the MERS coronavirus, it is again camel. And this one is again, a little bit disputed, but still believe that it is actually pangolin, which is what the intermediate host. So we have contact with these three intermediate hosts. So it is thereby human beings are getting infected. And during this, this kind of transition, because the species barrier when viruses are crossing, definitely it will adapt with a lot of mutations in that, where it can actually infect with the higher rate of infection the human beings actually. So let us see a little bit about science of that. I mean, basic molecular aspects of this virus, SARS coronavirus structure and replication. So this virus, as I told, positive strand RNA virus, you can see there virus has a surface. There is an S protein. S protein is a surface protein. Actually, this is the protein which actually interacts with the host cell while infection. And you can see that there is a nucleoprotein complex and then a matrix protein. And you can see the, it's a single stranded RNA inside this. So this S protein is actually studded in an envelope of lipid. So that's what we are calling, using soap or detergent and all, we can dissolve these things. Okay. Now, anyway, so when the receptor for this is actually angiotensin converting enzyme 2, which is there in the host cell surface, where this virus binds and actually infects it. The S protein actually interacts with the surface, receptor. This is ACE2, angiotensin converting enzyme 2. And once it is binding here, there is another protein. There's a protease is there in the surface, and that protease is a serine protease, which chops the S1 into 2, and S1 and S2, and finds its way into the cell, and through the fusion in the endosomal membrane where acidic pH is there. And after fusion, this whole thing, I mean, the genome is actually released into the cytoplasm of the cell. So this is the basic pattern of any, most of the positive strand RNA virus, especially flaviviruses and all. So then once the genome is actually ejected into the cytoplasm, the next stage is actually translation. So because it's a positive strand RNA virus, means it's just like an mRNA, and it can actually directly undergo the translation. There are, two major ORF1 and ORF1B is there, and these ORF1A and B actually, the next stage, the protein gets cleaved and it makes a RNA-dependent RNA polymerase and the protease of the coronavirus, and which is actually finally making a replication complex inside the cell in the membrane vesicles. Actually, membrane vesicle you can see here, and in this membrane, the later half, second stage, the remaining ORF actually codes for the structural protein. Whatever we are seeing in the mature virus, these proteins are actually translated from these region, and those proteins, you can see subgenomic mRNA here, which codes for different kind of different structural protein, and finally gets assembled here and exits from the cell, and once it is matured. So this is the process, and in the past, in the case of dengue and many other flaviviruses, dengue as well as West Nile viruses and yellow fever viruses, a lot of effort has been made to develop antiviral drug targeting each and every step here: entry level, protease inhibitors, and assembly, maturation level, and each level biochemically. Especially being a biochemist, always I will get excited by seeing this events. So this is the point where basic science can do a lot of things to dissect out the mechanism, how actually it replicates, what are the why host cell factors are required, can we interrupt that and then develop drugs against the viruses. These are the very interesting areas. And of course, a lot of drug molecule has been screened targeting dengue viral protease and RDRPs and RNA-dependent RNA polymerase. The importance is in the host cell has DNA-dependent RNA polymerase, but virus has RNA-dependent RNA polymerase, because virus doesn't use host cell polymerase. Instead of that, virus uses its own polymerase, that is RNA, because its RNA is its genome, so it's make negative strand, then again make a lot of copies of positive strand. So in this, there is no intermediate of DNA, so it never goes to the nucleus also, and everything is completed in the cytoplasm and it exits from there. Of course, there are some protein may be moving to the nucleus for regulate the gene host cell gene expression and all, but it is very, very interesting. And now coming back to some of the important question here today, what I would like to discuss with you. So if you see this global map, actually geographical discrepancy in the death rate of COVID-19. So now if you see this, it looks like, forest fire actually. Wherever firewood is there, it will catch fire. So you can see that a lot of red spots here and there, and in some places it is dense, some places it's less. So there is a clear-cut geographical discrepancy in the death rate of COVID-19. So a lot of factors are there. Of course, it is quite debatable. There are testing deficiency. Of course, every day we are seeing in the media and everywhere, testing is less, testing is less. So testing deficiency, of course, it will give an underestimated number. And age factors also, age factors and comorbidities are another thing. People are telling, the more death is because of the comorbidity and age factors. And sometimes we feel age is only not the matter, and there are other things also. And medical preparedness, that exclusively depending on the authorities, administrative authorities and governments and all. I think all countries, they do their level best to prevent this, no doubt in that. So medical preparedness. So genetic variation of SARS, variation in between, we have seen a lot of reports coming. There are some virus which comes with the more newer virus, and in the newer, in a disease with the more aggressive in nature. And that all things we have to actually look very, very in detail actually. That's about the viruses. So now coming to the case fatality rate versus recovery rate, if you see, you can see the geographical variation of case fatality rate is huge actually. If you see here, USA, the recovery rate is 18.7. This paper, in fact, it was published from our university by Professor Tangappan from Public Health Department. So it's a very informative article where he wrote USA, if you see the, the case fatality rate is 5.52. And you can see that in the highest in the case of France, the case fatality rate. And if you go in the other way, it's compared with our own state, Kerala. Kerala is actually case fatality rate. If you see 0.6 only, it comes. I mean, when you compare the other places, it is far less than the national average, that is 3.35. And recovery rate, I mean, in Kerala, it is, I mean, the highest, that is 96.2. I mean, then 94 is for, I mean, China, that's reported. I mean, so this you can see that a huge variation among the geographical region. If you from USA to, if you compare along with the other countries, and finally our own country, India. So there are a lot of difference. National average case fatality rate is 3.35. So what actually what could be the reason here? I mean, of course, it is again debatable. I'm not telling the only one reason or two reasons, many are there. But one thing is important thing today, what I would like to share is about the immune enhancement. Actually, that is one of the host factor. I mean, host interaction. So the key question is, what about the geographical variation of host immune factors? Who is host here? Host is a human being only. The host immune factors are different. I mean, it varies from place to place. And how does it varies? And the host immune factor itself is variable based on many other conditions, prevailing in its surrounding. How does the host immune factors affect the clinical outcome of COVID-19? That is another important area. I mean, we have a lot of, research work and knowledge based on SARS-CoV, which happened in 2002, and MERS in 2012. So again, why there are, in this time, I mean, I would like to bring your attention to the vaccine development. And I think around more than 100 candidate vaccines are underway now, clinical trial, waiting for clinical trial. And the thing is, we also have a lot of debates. I mean, like the vaccine will be ready by October, vaccine will be now ready by December, and a lot of news as well. Of course, we are also expecting that to happen with the positively. So meanwhile, the one basic question is why there are many unsuccessful efforts in the development of drugs against many emerging viral infectious diseases such as Ebola, dengue, chikungunya, Zika, SARS-CoV, MERS-CoV. The SARS-CoV happened in 2002, and again MERS-CoV in 2012. Of course, the main thing is we all are working on, entire scientific community is working on that. The thing is, it needs a lot of time actually. The average vaccine development time, I mean, I read in an article, is actually around 10 years, nearly 10 years after passing through the basic, I mean, safety, efficacy, and all. So there are a lot of reasons for the non-availability of vaccines against all these viruses which are emerging and re-emerging viruses. So if you tell about the emerging viruses again, I would like to tell that about the Chandipura virus. Chandipura virus is it was actually detected in 1960 in Chandipura in Nagpur actually. So at that time there was in India chikungunya as well as dengue was prevalent. So it was isolated in the fever patients. I mean, and then later it took at least 3 decades to again the virus again after 3 decades it emerged by causing the acute encephalitis in some children in Andhra Pradesh at that time. So then these 3 decades it was there, I mean, dormant, and then again, and of course chikungunya you can tell. I mean, chikungunya again it was in the 1960s and 70s. It was there. I mean, it was reports are there. I mean, again it took a couple of decades to come again, hit very badly in 2006, especially in our own state. So now it is actually contained, and we are not contained in the sense we are not seeing that many number of cases. But dengue is another thing. It is actually repeating, and again in that dengue we know that 4 different serotype, and each year when we see sometimes some area one will be there, another will be dengue 2 will be there. There are lot of variations we can see across the country. And similarly, so there are so many hurdles in the development of vaccine also. Let us, before going for the immune enhancement, let us see what happens when SARS-CoV-2 infects in the lungs actually. So the inflammatory response of SARS-CoV-2 infection, we can see here the is a primary barrier. I mean, actually there is epithelia or all aerosol which will enter into the lungs and the trachea, and then finally the epithelia is actually coming into contact. So when coronavirus 2 infects, the activation of, we can see the basic immunology, that is the macrophage activation and dendritic cell, T cell activation, and finally leading to in the case of coronavirus 2, this actually induces a lot of pro-inflammatory cytokine. The reason for the clinical outcome is because of the pro-inflammatory cytokine. So it induces fever, it induces impaired hematopoietic functions like difference in the count, blood cell count, and disseminated I mean, there is a lot of vascular biology to play here in this actually inflammatory response. And serum protein reduction, like that, finally kidney injury and so many downstream effect we can see. This is what the place where actually the patient will become, the situation will become fatal. And it can be classified into two different: primary inflammatory response and secondary inflammatory response. I mean, when the first stage is actually the normal apoptosis and pyroptosis. Pyroptosis is a way of cell death. Apoptosis is also cell death, but with the pyroptosis is purely result of inflammation, and pulmonary cell infiltration will happen and leading to the fluid accumulation in the alveoli. And definitely we know that when fluid is accumulated in the alveoli, we can see that there is a decrease in the oxygen concentration in the blood, and then decrease in the oxygen, then naturally the blood pressure also will shoot up, because then that also is another important reason in this. And another important area is the antibody actually binds to the FCR receptor, antibody receptor. The antibody, you can see here, antibody binds with the virus, and the other end of the antibody, that is FC region, binds with the receptor of the cell which is infected by the virus. So then once it is bound, this can go in different ways actually. The outcome of this complex is having a lot of role in the disease progression and clinical outcome. There is actually, you can see that the macrophage can actually, M2 macrophage can actually secrete MCP1 and IL-8 production, acute lung injury, cellular damage, and macrophage response itself is changed. You can see the complement system activation, and antibody dependent cellular cytotoxicity also. It's based on natural killer cell will come into play. I mean, a complex events will happen in the secondary response, where totally it is an inflammation and leading to the alveolar injury and edema. So now coming to, just I'm comparing for a moment with the inflammatory response due to dengue infection. So this is what SARS-CoV-2 I told in the lungs. Actually, in the case of dengue, the infection happens in the skin because of the mosquito bite, and there also you can see all these cells actually. Finally, the macrophages, dendritic cell, skin resident dendritic cell, actually, and the CD8 cytotoxic cell and natural killer cell. These are all actually activated, just similar to the other cases where lungs, in the case of COVID-2, here also you can see the immune-mediated injury leading to the IL-8 and IL-6, interferon alpha, TNF alpha, interferon gamma, and this is leading to the vascular leakage and finally becoming the fatal situation. And you can see the similarity. This is what I was telling. There is a similarity in the clinical outcome and the molecular events and immunological events involved in the disease progression also. So now let us have another important aspect of this. Where if you see around 50% of the global population is under risk of dengue. I am just comparing, I'll come back to COVID. So the 3.6 billion people at risk for dengue. And in that, we are getting out of total infection, around 390 million infection. Out of that, 75% are asymptomatic infection. The same thing what we are seeing in the case of COVID, where majority of the population have asymptomatic infection. And in this 75 asymptomatic and 25 is mild symptom like fever, like febrile illness. And that is 25%. These 25% from this, a few of them will be hospitalized. Finally, 1 to 5% of severe cases, which is resulting. So this 1 to 5% is actually, I mean, if you compare the asymptomatic infection in the COVID also, you can see that the severe cases are in less, but it is not the same across the country. So there is a lot of variation. So as far as our own state is concerned, the symptomatic cases are, I mean, severe cases are relatively lesser. And here you can prevent vaccine can be used here for as a prophylactic, and we can do vector control. We can do directly acting drugs here to reduce the viremia. And actually, finally, once it is serious, of course, we have to do the symptomatic treatment. I mean, symptomatic approach for the management of the clinical management. And why this? Some people are getting more, severe cases. Some people are less severe. In the case of dengue, is somewhat better explored, where this is what the area called antibody-dependent enhancement of dengue viruses. Or the antibody-dependent enhancement in the sense if a person is getting infection with one particular serotype, if he gets a successive infection with another serotype, like I say, a person infects with the dengue 1 and he gets a dengue 2 infection, there will be cross-reacting antibodies between these two serotypes. And this cross-reactive antibody will make immune complex, but it won't neutralize. Instead of that, it will enhance the uptake of the viruses into the cell, into the monocytes or immune cells. Because the immune cell has a FC receptor. That is, FC is the antibody region we are seeing here, constant region, which has a receptor here. So this complex will be taken inside and there will be enhanced viral replication in the case of dengue. So this leads to the hyperinflammatory sequence and leading to the severe. In the case of dengue, it is not. It's a complex. The reason for this actually, it will be a little bit more clear if you have a look on the envelope protein of the dengue. There is an envelope protein. You can see this here. There are distinct what we are calling. One is of a host receptor binding domain, that is a domain 3, and domain 1 and 2. So this domain 3 is type-specific neutralizing receptor binding domain, and which actually induces neutralizing antibody, whereas this area is non-neutralizing. But this entire protein you can see on the surface of the virus. When this virus infects the human being, infects us, actually it will create a polyclonal response, and which will be antibody will be targeted to all possible epitope which is presented by your system. So then what ended, some of them are neutralizing, some of them are non-neutralizing. Neutralizing will, of course, is a good ligand, put it as a good antibody, and it is actually neutralizing and helping. The other phase is non-neutralizing antibody will make immune complex with another heterotypic infection during the heterotypic infection and immune complex, and it will enhance the uptake. That's what we are calling, in other words, immune enhancement. So now I would like to bring your attention to the dengue vaccine. So we know that 1 year back we have seen that there is a new vaccine. Introduced. They were trying to test in the human population, clinical trial phase 1 and phase 2. They were trying. That is the dengue vaccine by Sanofi, Aventis group, and unfortunately it was withdrawn after some time because the reason was it was not working in the seronegative people. The people who had already got an infection with the dengue, it was not causing any problem. But the people who are naive, they didn't have any infection. Those people started to cause certain clinical manifestation which was severe. So that's why it has to withdraw. And in fact, in India, it was not actually tried also dengue vaccine. And in Brazil and certain, around 20 countries across the globe, they gave permission and they did the trial. But it was later on data. Currently, the situation, we don't have any vaccine for that. So here again the reason, the attributed reason is immune enhancement for the dengue also, and causing the severity of the disease. As I told, it is making an alternative route to the entry into the macrophages and then resulting in the enhanced viremia, and this results in the hyperinflammation. And now I showed in this, in this slide, the two important vaccines being developed in India is actually one is tetravax dengue vaccine. It's actually licensed from NIH, and this I think I'm not sure in what stage the trial is, but this is a live attenuated virus where the genome, you can see these three boxes is indicated here, and this box indicates that there is certain nucleotides are deleted. So this deletion makes it attenuated, and these are mixture of 4 different viruses which has complete structure of envelope, and all complete structure is there in this. It's a live attenuated vaccine, and is basically from NIH license. And another thing, what I would like, the second one, ESV4, is actually completely this has been used domain 3 of the envelope protein, which I have shown you in the previous slide, is neutralizing it now. And other parts have been trimmed, which can induce the cross-reacting non-neutralizing antibody. So this domain 3 actually made a primary construct where 1, 2, 3, 4, 4 serotype, and expressed in the Pichia pastoris and along with the hepatitis surface antigen, and it makes a virus-like particle, like a mosaic model. I mean, a virus-like particle is being working in the ICGB New Delhi, where actually this asset, I was actually involved in that as a postdoc in the beginning, and using the delivery of this asset, antigenic asset, into the using the adenovirus vector. In fact, the same adenovirus vector being explored for the COVID vaccine now, I mean, where you can see the S protein of the COVID is actually, grafted into the adenoviral genome and used as a vaccine candidate, and also being tried as a vaccine against COVID-19 in UK. And this is what DSP4, this is what, ideally this should not cause any enhancing antibody in this case. But here again, it is your suspicious non-neutralizing epitope will be there. Now coming back to based on this knowledge, I mean, I was just looking into the literature. We can see that this article came in Nature Review in 2022. Actually, you can see there is a desperate need of effective therapies and vaccines for SARS-CoV-2, but the vaccines are being developed at an unprecedented speed, and are already in clinical trials without doing clinical testing for safety and efficacy. This is what one of the important things what we have actually looked into. The safety and efficacy, of course, we all need vaccines very fast, but how better we are able to do the safety trial. Some of the time, I mean, as I mentioned in the beginning of my slide, I mean, we were having coronavirus infection as a common cold. Definitely, I think the population might have already immune response or antibodies against certain epitope of the coronavirus possibly. So here the question is, how in that dose response this antibody is going to respond in the vaccine candidate? And vaccine is being developed in both the traditional as well as in the modern platform, is the recombinant vaccines, subunit vaccines and all. So here things need to be watched and more scientific. What I am telling is more scientific research is needed in this area, more academic research is needed in this area. Now again, you see another thing is COVID-19 receiving ADE from other coronaviruses? So this came in another important journal, I mean, very recently in the February. And other coronavirus, what I mean is, of course, I have shown you that family of coronavirus, what is there prevalent in the surrounding. So whether it will cause, we don't know. We need to be taken into account. So if you see this is Janus phase, I mean, actually two-headed thing. Antibody is good and antibody same, antibody is playing bad. So outcome of antibody response. Here you can see this is a typical coronavirus in the thing, and bound with antibody. It will bind the neutralizing antibody. It stopped here. This story stopped here. This opsonization degraded and is fine. And now what happens is a non-neutralizing antibody finds its way into the macrophage and actually enhancement of multiplication causes again TLR3, TLR8 and innate immune response and leading to the activation of pro-inflammatory cytokine and leading to the severity of the cases. So how some of the vaccines are really going to work? Of course, I'm not telling all vaccines are bad or something. Please, don't take that way. I'm telling need to be studied more scientifically before introducing to the human population. And you can see the good and bad thing of same thing again. Outcome of antibody response. Here we see the ADE, what I already mentioned, binds to the receptor, enhances the ADE is happening, antibody-dependent enhancement happens, and virus exists. At the same time, if it is binding through the FcR receptor, and you see the M1 macrophage is there, and it is actually activating the other kinase pathway, and finally leading to the inflammatory cytokine release, and this is dangerous. This hyperinflammatory response will happen. So meanwhile, there is a target for here. I mean, probably this can be targeted by using the drugs or some blockers for which causes the activation of this kind of hyperinflammatory response. So therapeutics. So negative regulation is there here. So we can Fc gamma receptor we can be targeted by antibody or other some downstream with some small molecules, and where we can actually control this activation of this unwanted things. So you can see here one more important paper. It came in Journal of Virology very recently. They actually explained the structural elucidation they have done, molecular mechanism of antibody-dependent enhancement of coronavirus into the cell. So how it happens, what they usually, the S protein of the coronavirus actually gets proteolytically cleaved by the action of host cell protease, which is present on the cell membrane. And then once it is stopped, then S1 actually interacts with the angiotensin converting enzyme 2 receptor. So in the case of ADE, what happens, certain antibody actually binds to the S protein on the surface of the virus and makes and induces a structural changes in the S protein. And these structural changes actually facilitates the rapid proteolytic cleavage by the host cell protease to the protease which is present on the cell surface, and thereby finding its way, enhanced way of entry into the cell in a normal receptor itself, not through Fc gamma receptor. Immune complex actually binding of antibody makes the S protein more prone to the proteolytic cleavage. So then the uptake becomes faster. So this is what this came very recently, and again COVID-19 has Janus phase of immune enhancement. So we need to actually find out these, whether really happens this. And bringing your attention again, so this is true for the antibody-dependent enhancement also found out in the Ebola virus infection, where human antibodies isolated from the survivor. So after Ebola virus infection and some survivor was the isolated antibody caused the enhanced uptake of Ebola virus. There are reports actually. And of course, dengue is quite well known. And another important thing is Zika infection. Zika and the dengue virus, they are flavivirus, both are flavivirus, and some of the antibody even it is cross-react. In our normal lab protocol, it is cross-reacting. So the pre-existing immunity, pre-existing antibody for dengue may actually affect badly for the Zika infection, probably Zika related. This is also a very important observation. And dengue Zika virus co-infection. So now coming to another important paper. So whether it is really so here you can see the one important study came in ACS infectious disease, where a certain S protein derived, coronavirus surface protein, S protein derived protein-derived peptides was used as a epitope. Then they actually explored whether these peptides can induce ADE. Of course, they found one of the peptide. You can see here. I think you can see here. Yes, 597 to 625 peptide in that region. It causes antibody-independent uptake in the cell. This work was done in RHESUS macaque, is a non-human primate model they have done, and whereas the other epitopes, other peptides like S471 to 503, these couple of peptides, they induced ADE. The antibody against this particular sequence epitope induced antibody, induced immune enhancement, whereas some other peptides it was not inducing. Instead of that, it was neutralizing antibody. You can see that neutralizing function is written here. So we can see clear pathological consequences here. In the infected macaque, this is actually vaccine challenge experiment was conducted in the RHESUS macaque where they immunized with the formaldehyde attenuated SARS viruses, and then did a booster at 14 days, and then again challenged with the live virus in the nasal cavity inoculation. They actually challenged, and what they found is the ADE happens in the RHESUS macaque, where actually you can see my pointer is not working here. Sorry for that. We can see the second one, ADE macaque. You can see that alveolar infiltration and edema, whereas protected macaque, the antibody which they have used actually with the sequence specific to the not neutralizing antibody, it is protecting. Other one is inducing more damage. You can see the edematous lungs there. It is a very important study. And coming to in this stage, I would like to explain some of lab work. What we are doing in our lab here is actually we in our lab we are using PHP1 cells is a monocytic cell line, and we are looking actually whether adenovirus vector, as I told, adenovirus is a vehicle, actually it's a vector used for vaccine purpose, and even COVID-19 vaccine also people are using vector for it. Actually there are different variety of derived vectors from adenovirus. This is classically used by adenovirus serotype 5 vector. What we are actually looking for is whether this adenovirus can induce ADE in the normal in vitro lab setting, and so can we measure that? So what we did is actually we took two different kind of cells. One is the normal cells, that is the normal cancer cells. Other one is monocytic cell. This monocytic cell doesn't have PHP1 cells, it doesn't have any receptor for adenovirus, whereas HeLa cells has a receptor. So the moment you put the adenovirus expressing a reporter green fluorescent protein expressing protein, you can see dense expression of GFP, because more viruses are entering into HeLa cells because it has normal receptor for the adenovirus serotype 5. But the PHP doesn't have a receptor and it won't infect. However, if you pre-mix the virus with the normal human sera, normal in the sense not, without any clinical symptom at all. Those sera when we take and make immune complex and add into the monocyte, it is taken in a more enhanced way. So you can see in the next slide, this is how the HeLa cells work. If you increase the virus concentration, there is a linear regression. You can see it's linearly correlated with the fluorescence and infection. The moment you put antibody, with that, in the HeLa cells it will neutralize, it won't enter. But if you put it in the other way, when you put it in the monocytes, you can see here is only adenovirus when you infect, nothing happens, it is not entering. when you mix with this is a study we have done, at in ICV where murines immune sera was used to make immune complex with adenovirus and infected. Then we have seen, pre-immune sera means no antibody. Then there was no entry. But when we mix with the immune sera, means my sera, which is having more antibody towards adenovirus type 5, it rapidly enters into the U97. That is another monocytic cell line. And recently in our lab, we have done the same thing with the DHP1 cells. And this is murine sera, we have used the human sera, and we can see a clear correlation, at a certain dilution. This is the serum dilution. At a certain dilution window, the uptake of the virus is more. That means antibody, the presence of antibody at a subneutralizing titer probably also bad. So immune enhancement can happen with the non-neutralizing antibody and also non-neutralizing antibody and also neutralizing antibody probably the sub-neutralizing level. So these are all the matters. I mean, you can see here enhanced uptake in a particular dilution. Whereas if you do another human sera, it will be needing more dilution. It will enter here. So this we have screened around more than 68 people. What we are finding is the uptake of. But this is a model system to make. I mean, to make it more clear, this is a model system to study how immune enhancement happens for the virus or the enhanced entry into the cell. So when we do the further dilution, probably, I mean, in some other individual who has more titer, maybe we need another window of dilution. So these antibody responses it is actually varies from person to person. So we can't make a generalized, dosage of the virus here to know the entry and all. So that's what, it's highly variable. So now, this is now currently what we are doing is, can we use this platform? So the immune complex enters in the monocyte. So can we block, can we block the entry of the immune complex conjugated with the RGFP into the cell? By a small molecule, actually we are using certain kinase inhibitor to know whether it is really inhibiting the entry. So all inhibitors of entry may not be clinically useful, but to understand the basic science, we need to know what are the type of receptor. It may be FC receptor, means it may be CD16, it can be 32, it can be 64, and if so, what are the downstream kinases involved in the activation of the uptake of the virus into the cell, and whether it can be generalized to the other virus, and it may be because host system is saying only virus varies. So virus doesn't induce, the virus doesn't contribute a new factor for the whatever existing system in the host cell hijacked by the virus. Viruses are very economical. It won't carry big argument. It actually modulates the host system. So maybe some of the conserved mechanism may be exploited by multiple viruses. That's why we are telling about pan inhibitors of the virus. One inhibitor can be repurposed for another virus. That's what repurposing of the drug also very important thing. Similar to the vaccine, the way the vaccine trials and things are going on, similar to that, the antiviral drug discovery also very important at this time, targeting multiple non-target, and a lot of repurposing work can be done. And this work actually supported by KSCSTE fund only, this current work what I have shown. And finally, I would like to conclude in that, I think it's 4:30. So we are just beginning to understand the dynamics of COVID-19 in 2 months and the impact of the virus on the individual. Lot of academic and scientific research, focused scientific research and baseline data is needed for understanding the host-specific immune response and clinical outcome. That is very important before even introducing the vaccine and how this vaccine. Preclinical work, a lot of work is needed before introducing into human being. And immune enhancement or ADE need to be considered while designing vaccines against COVID-19. So the reason is the stories, whatever I was telling till now, like the dengue and whatever you take, there are this component is there. Nobody is actually there are a group, two school of thoughts are there. One school of thought actually they don't think it, they think all it is in the laboratory level thing. Another group of thought actually is telling actually there is a phenomenon, it will cause issues. So, but it is better to have a very safe platform before embarking into the vaccines. And in fact, in our lab, we developed an effective method for estimating ADE of high antibody EFE, and this can be using a fluorometric. We can make it a high throughput assay. So this can be one of the platform what we have developed to screen the small molecule drugs to understand the ADE. Finally, I would like to acknowledge my thanks to this big team. I mean, our COVID-19 test team of Central University of Kerala. Probably, I think probably in our country itself, maybe among universities, I think our university is the first one to start COVID testing in the department, and that is mainly because this team whom I am displaying here, my colleague Dr. Samir is there, Dr. Shirakaman is there, and other research scholars, Ranjit, Lalita, Lindsay, and Pravida, Prajit, Vishnu, all people, some of these people, they are coming in and out, and I mean, they are doing a lot of work. I am talking to you here now because they are in the lab. Otherwise, I would not have even spent 1 hour. So, a lot of sample loads are there. So, I give my acknowledgement to my COVID-19 test team, and also our university, Central University of Kerala, and also our Vice Chancellor and other university authorities. And our Vice Chancellor, Professor Kumar, has given a lot of support in for doing this COVID-19 test in our lab. And also, I would like to acknowledge ICMR, RTPD, and KSCSTE and all for giving funding for our basic research work. This is our campus. Thanks a lot for hearing.
Thank you, sir. Actually, it was really wonderful to hear your talk. It was really informative, right from the explaining the origin to the recent research happening at your lab. Thank you very much, sir.
Thank you.
I think now we can take a few questions. People have posted the questions in the chat box. The first one is a little general, like is virus that created chaos ever in the history? I think just he wanted to know about the viruses that created chaos ever in the history, like maybe man-made or natural.
Yeah, that's what I mean. This is a basic question of evolution. I mean, the thing is, viruses really don't want to create chaos. It actually trying to find out its own, host and survive. The basic tendency is to survive for every, or in so many virus, we can't call it as a living organism, but what I am telling you is it's a part of evolution, it defines it. And the creation of chaos, actually, we are also creating a lot of chaos in the nature, and then we are surviving. It's the basic principle of thermodynamics itself tells that during our lifetime, we are actually creating chaos and we are moving ahead. So, of course, the evolution happens from, that is the basic answer, I think. So, that is part of our history.
Okay, yeah, yes. So the second question is related to herd immunity, how it helps in combating such an epidemic, since it's a natural barrier.
Pardon?
So the next question is about attaining herd immunity against this pandemic. So the role of herd immunity in combating.
Yeah. Herd immunity, of course, we need to explore that more. I mean, as I told, there are a lot of gray areas in this. But one of the things I was talking about herd immunity only, the immune enhancement, which is a bad phase of herd immunity, when it causes actually, what I am telling is, let us say I am getting infected with the dengue 1 and then successive infection with the 2 or 3, then if it is protecting, it is good. herd immunity, but if it causes the severity of the disease in the other way by immune enhancement, then that also it goes in a bad way. Actually, herd immunity is there. As I told you, it's just a common cold viruses. SARS viruses were common cold viruses from 1960 onwards. We knew that. So definitely there will be herd immunity, but I don't know whether it is protective enough. And another thing, how long the herd immunity memory is, sustaining in the body, memory response. How long this memory will be there in the body, that also long-term immunity it should be. So that also we are not sure.
Okay. So the next question is related to the plasma therapy. And is there a chance of ADE during the plasma therapy?
Actually, till now, I don't think any bad reactions or immune enhancement reports based on plasma therapy, probably because we are taking plasma from the survivors.
Okay.
Probably that may be one of the reason, but I was also looking into the literature. But safety is it's a part of clinical protocol now. Yeah.
So actually, what happens during this plasma therapy?
Pardon?
What really happens when a patient undergoes plasma therapy?
Plasma therapy, in the sense, survivors will have antibodies which can neutralize the virus. So, the survivor's plasma we can actually inject into the patient where it can potentially neutralize the infection of the virus.
Okay.
Yeah.
So there's another question related to so plant-serving protease inhibitors have been isolated from several plants, including Moringa prolifera. Can we use inhibitors to neutralize the serving protease and thereby preventing the viral entry? Yes.
So plant-serving protease inhibitor, right? You are told.
Yes.
Yeah, definitely that need to be cleaned. Actually, that's a good process of work. Actually, there are, as I mentioned in my slide, there are a lot of points or area where we can actually target this infection and related downstream process. Like one of the thing is entry level itself. There is serine protease, which is actually cleaving the S protein. Probably if we can inhibit that, it will really maybe we are getting a good drug. It will be very good actually, natural product. Until we do the wet lab work, we can't tell. But because by my experience, we were doing NS3 dengue virus protease inhibitor screening, and those inhibitors were used to suppress the replication of the virus. When we do the live virus infection, it used to suppress. And but however, I don't know, many of these drugs have been not coming into clinical practice. There are a lot of millions of molecules which are very screened in this area as protease inhibitor against viruses, and either because of the toxicity, when you are going to the real clinical setting, it will that is the one of the reason. Otherwise, there are a lot of efforts and a lot of molecules already being explored. But plant product will be actually. I can quote today morning Professor Subhash gave a wonderful talk on the antiviral drug used luteolin. It's a plant-based luteolin, and it actually inhibits furin proteases in the host cell. Furin protease is required for the virus protein processing, and so if you suppress the furin proteases, virus cannot multiply. So luteolin, it is an important. So like that, a lot of works are there. Meanwhile, the problem is these kind of drug when we add, virus will generate escape mutation. It will escape from that. It will create escape mutants, and definitely it will adapt with a newer situation. So that's what the evolution of virus also happens. The moment it gets a selection barrier or selection pressure, virus tries to overcome that by creating more mutation in this, because there is no proofreading machinery in this. So this is a good thought. These actually need to be explored.
Okay. Okay, sir. I think the next question is regarding the analysis of samples which you are doing in your lab.
Oh, okay.
What extent of error is there in analyzing the samples? It is heard that unless the sample is from a symptomatic patient, there is a chance of getting false positive. Is it true?
False positive. I mean, which test they are referring? RT-PCR or antibody test?
Okay, that's not mentioned.
Okay, anyway, I think they are mentioning about the RT-PCR. What we do is RT-PCR test.
Both. For both tests.
And antibody, there are chances of false positives are there because we compare that. I mean, antibody positive when we do the RT-PCR test many times becomes negative. So I mean, so the same way, the RT-PCR error in the sense, of course, there may be error. I'm not denying that, but it is more sensitive. And no need of any symptom and all. I mean, it is actually when there is a viral genome, even very less copy number also it will pick it up very sensitively. And we do multiplexing, single tube multiplexing PCR, RT-PCR essentially. It is not quantitative; it is a qualitative assay. And we currently check envelope. I told you envelope protein and S protein, S gene expression and E gene expression. In the throat swab, actually, we check throat swab. Throat swab, we will isolate RNA and then we will make do the PCR by multiplexing.
Okay.
Yeah.
So actually, are there studies that people with autoimmune disorders and the effect of coronavirus on the people with autoimmune disorders? Like, are they highly prone to death, or is there any study regarding that?
I didn't come across, it's a good question. I didn't come across. So I can't answer. I think, I mean, depending on the autoimmune disorder, it may be, I mean, it is again a morbidity issue. So, depending on the autoimmune, which organ actually, there are many autoimmune disorders. So, which specific organ is antigen is targeted, autoantigen is targeted, that probably may be the one of the deciding factor to do some analysis in that direction, I believe. Yeah.
Okay. So actually, I was just reading one article from the newspaper that when such a pandemic happened or broke in India, most of the people's concern were about the people living at crowded cities and especially in slum areas like the slums in Mumbai. But even though what I read was the number of cases in Mumbai is much higher than in the slums. The people are staying or living so close. And one of the reasons they are mentioning is that the number of cases being reported is less because usually the checking process is so stringent that they try to analyze the samples from all people who are showing some kind of illness. So is it the only way? Like, I mean, unless herd immunity is, as you said, I mean, we cannot wipe out the last virus from here. I think we need to live with the virus.
So there is one thing is there. I mean, there is Harold Warmus, who was an NIH director who got Nobel Prize for the oncology. He actually wrote that. I mean, it is actually in that. What we can feel is virus is like what we are calling, Srishti Sthiti Samhara. All these things, virus is actually a very powerful thing. I mean, we can't wipe out probably entire virus. Last virus I can wipe out and go home. I don't think it is feasible. We have to live with the adapted situation. Yeah.
And we need to continue this regular checking process until a vaccine or all other people get vaccinated. Or
care need to be taken. Otherwise, it will be a problem. I mean, care is very important. I mean, finally, it will be part of our life. I mean, we won't feel difficulty in following these directions. Until unless we are getting effective vaccine.
Yeah. Okay.
Yeah, and antiviral drug. See, there is another, I don't know whether, see, now many people got infected with the SARS-CoV-2. We don't know the same virus, how it is infecting the animals, here, and what way it is going to, replicate there. These things also is an important concern.
The secondary infection.
Yes, to the animals, and then how it is going to actually affect in future. That need to be studied. That's what I was telling. Is constantly we have to follow up scientifically.
Okay.
That is very important.
So, in your research lab, what will be your immediate focus? So, what is the next focus?
Actually, I am not actually working on research. I was not doing any SARS coronavirus. Only now, because of we were doing these are all positive strand RNA virus, as I told, and based on the RT-PCR, we can do diagnosis. We thought at the time of lockdown there was no classes and all. We thought we can actually dedicate our lab by doing this. Because it's Kasaragod, we don't have much facilities. Here there is no medical college, and so we actually adapted our lab for that. And next task, of course, we got a research grant from BERAC, and where we are going to develop a test kit in collaboration with a company that for developing. It's called COVID test, RT-PCR test kit using saliva.
Okay.
That's one small grant we got. And of course, I would like to explore one possible thing is what is the level of seroprevalence in the population using neutralization assay, virus neutralization assay, using pseudoparticle in future. That I want to check actually.
Okay.
How much level of, because not the carbon test what we are doing, it is a in vitro laboratory based neutralization assay we are planning. And it start, once our testing should complete, 24 hours we are working for testing now.
Thank you, sir. Thank you for
very much.
taking such time to give us a wonderful talk. We really know how busy you are. Thank you very much, sir. On behalf of the PhD and all the participants who have attended the webinar, I express our sincere gratitude to you. Thank you very much.
Thanks a lot.
Thank you all participants for joining us for the webinar. Thank you very much.
Thank you.